When starting the replication process, helicases meet the DNA at the origin of replication and begin to separate at the 5 and 3 strands of DNA, making them available as template strands. Helicases untwist the double helix at the replication forks and the parental strands separate. This creates single-strand binding proteins. Enzymes that synthesize DNA cannot initiate the synthesis of polynucleotides. They can only add nucleotides to the end of an already existing chain that is base paired with a template strand. As each monomer joins the growing end of the DNA strand, 2 phosphate groups are lost as a molecule of pyrophosphate. The initial nucleotide chain that is produced during DNA synthesis is actually a short stretch of RNA, not DNA. This RNA chain is called a primer and is synthesized by the enzyme primase. Primase synthesis RNA primers using the parental DNA as a template. Enzymes called DNA polymerase catalyze in the synthesis of new DNA by adding nucleotides to a pre-existing chain. DNA polymerases can add nucleotides only to the free 3 end of a primer or growing DNA strand, never to the 5 end. Thus, a DNA strand can only elongate in the 5 to 3 direction. DNA pol 3 remains in the replication fork on that template strand and continuously adds nucleotides to the new complementary strand as the fork progresses. The DNA strand made by this mechanism is called the leading strand. To elongate the other new strand of DNA, the mandatory 3 to 5 direction, DNA pol 3 must work along the other template strand in the direction away from the replication fork. The DNA strand elongating this direction is called the lagging strand. Then, DNA polymerase 1 replaces the RNA nucleotides of the adjacent primer with DNA nucleotides. This results in the 5 end of the DNA fragment to be added. An enzyme called DNA ligase joining the sugar phosphate backbones of all the Okazaki fragments. Since DNA polymerase 1 cannot join the 3 end of the fragment to the 5 end of the Okazaki fragment, the DNA ligase bonds with the 2 together to make one big DNA strand.
This shows the lagging strand beginning to form what is known as the trombone model of replication. Due to the fact that the lagging strand has many more steps and enzymes associated with replication, this trombone-like structure helps orient the replication enzymes for repeated synthesis. Once DNA polymerase 3 reaches the RNA primer of the next Okazaki fragment, it will detach and begin synthesis of another fragment.